Right now, every second, approximately 2 million cells in your body are dividing. That means approximately 2 million cells are going through the intricate steps of mitosis, creating genetically identical sister cells that are able to produce all the proteins your cell needs to function. You can imagine how complex this is. It takes the effort and interaction of countless proteins to regulate mitosis and to ensure that cell cycle is not unregulated. One of the proteins that are of particular interest to us when it comes to cell cycle regulation is a protein called SCIP2. SCIP2 plays an important role in S phase of mitosis and helps degrade regulatory proteins and allow the cell to proliferate. Because SCIP2 helps promote cell division, it's been labeled as a proto-oncogene. This means that SCIP2 has the potential to lead to uncontrolled cell division and eventually cancer if left overexpressed. In fact, several types of cancer tissues show overexpression of SCIP2. However, I'd like to show through my research that SCIP2 plays an important role in cell cycle regulation, beyond just being a proto-oncogene. As it turns out, while overexpression of SCIP2 may lead to things like cancer, underexpression of SCIP2 or the loss of SCIP2 in cells also leads to polyploidy, which is another form of uncontrolled cell division where cells end up accumulating excess copies of DNA or chromosomes. In this sense, SCIP2 seems to play an important regulatory role in preventing polyploidy. Specifically, our lab believes that it's the interaction between SCIP2 and cyclin A, which is another important cell regulator that's important to help prevent polyploidy. Cyclin A and SCIP2 have been shown to interact in various mitotic tissues, and just like SCIP2 and its loss leads to polyploidy, the loss of cyclin A also leads to polyploidy, which is why we hypothesized this. In order to test our hypothesis, I will be conducting a series of experiments to help characterize the SCIP2 cyclin A interaction and in turn help better understand cell cycle regulation. Exploring how SCIP2 functions in the cell cycle will help broaden our understanding of mitotic regulation and can help contribute to our understanding of SCIP2 targeted treatments for cancer.